taste in that.
Because it works.
Yeah. So we have salivary amylase that works in the mouth, a little bit of the esophagus, but it's denatured in the stomach, and then the other organ would be the pancreatic amylase. Okay. Lipase is made by salivary glands and stomach. So this works in pancreas. All right. So question number 2. Which of the following molecules is a hormone?
Gastric inhibitory peptide.
So remember, if the molecule ends in ASD, it's always an enzyme. But ending in IN could be either an enzyme or a hormone, depending on the molecule. You can always go by that.
D.
Think about function. Think about what the chemical is doing to help you decide whether it's a hormone.
And B.
Does it act to change behavior, or does it act to increase chemistry and digestion technology?
What? Yeah. I have to take a picture of this. It's A and C. Or A and C.
One of them.
Only enzyme.
So what is the behavior of pepsin? What does pepsin do? Okay. It increases the reaction which functions to work on proteins, carbs, or lipids. Proteins. So take polysaccharides and increase the reaction of hydrolysis, which forms like 4 to 5, 4 to 6 amino acid sequences or dipeptides. Okay, what does gastrin do? So what cell makes gastrin? G cells. Which another name for the G cells are? Where do we find them? At the bottom of the gastric glands. Right? Another name for a G cell is enteroendocrine. So gastrin functions to increase activity of the stomach, secretory cells, as well as stomach muscle motility. All right. What is trypsin? What organ makes trypsin? Pancreas. Acinar cells in the pancreas, and it's actually released as trypsinogen. It's activated to trypsin. It works in the small intestine. It's an enzyme or a hormone? Enzyme. So it also is very similar to pepsin. It's going to take large proteins and break them down to shorter chain peptides. All right. What organ makes secretin?
Trypsin or enzymes. Pepsin and
Small
tyrosine
intestine.
enzymes.
Hormone or enzyme? Hormone. And gastric inhibitory peptide? Hormone. Okay, so we'll leave that up there. Well, remember I promised you I would come back and summarize what we ended our discussion with on Tuesday by
BDE.
drawing a diagram. Actually, I'm going to change the slide to the diagram. So we have the stomach. I don't think I need to label it, but I will. So you have it for your images.
So BDD.
I suppose BD because it's just like that when I'm going to bed.
All right, so we have the stomach.
You guys have
gene to show it's going to activate the vagus. Okay? Now, if you smell bad food or food you don't like, then it's going to activate the sympathetic system. Okay? And we'll put a smiley here. But if you smell food you don't like or you're already sick, then it's going to activate the sympathetic system. Okay? So when I was in high school, I don't know why we did it, but my older sister and I were like junior and sophomore in high school. We decided to throw a banquet for our parents. My sister went so far as to make a 2-foot tall statue out of paper mache of a Italian woman with a vase over her shoulder, and we had a room, a tiny, tiny little room about this big that we called the music room. There was only big enough for one chair and a piano. So we set up a card table in there with nice china and tablecloths and sent out printed invitations, hand printed. My little sister was really too young to help cook. And we made fresh bread and spaghetti sauce, and they led them to this Italian dinner. I don't know what we were trying to get out of them, but anyway, all three of us girls came down with the flu. So all I remember from that is lying around on the floor while my parents ate their fancy dinner. And for a year, anytime I smelled spaghetti sauce, I felt nauseated. So those kind of memories, that we hold with food and texture and stuff. So the vagus, if we draw a line from the vagus to the stomach, is that going to be excitatory to the stomach or inhibitory? Excitatory. So it's going to do both increase secretions of gastrin, and it's going to increase secretions of enzymes, and it's going to increase motility. Okay? If we excite the sympathetic nervous system, what's that going to do at the stomach? Opposite. It's going to constrict blood vessels, it's going to decrease constriction, and it's going to decrease motility. All right? We'll put an inhibitory there. And then, as I said, one of the activities of vagus is to the release of our previous slide hormone, gastrin. So vagus is going to increase gastrin. And does gastrin have an excitatory effect on the stomach? Yes. Okay, you kind of see the plan that we're doing here. All right? And then part of that stimulus by vagus is to relax the sphincter. So while it's causing contraction of the muscularis externus, it's going to relax the sphincter. And our acidic chyme, that's what we call the churned up food stuff mixed up with acid and digestive enzymes, that is going to move into the duodenum. Okay? So we had 3 hormones that we described being released by the duodenum. Remember I said earlier that the duodenum is really too short for any digestion to take place. Instead, it's a mixing receptacle for the chyme from the stomach, the bile salts from the gallbladder, and the bicarbonate ions and enzymes from the pancreas. So let's list those 3 hormones, and we'll do gastric inhibitory peptide. And that is going to be inhibitory to stomach. Just going to give a little pause while we add and mix up components in the duodenum. And we don't have any other function to assign it. And another one, there's 2 others. Secretin or secretin. And this binds to receptors on 3 different organs. So its origin would be the small intestine, okay? The endocrine cells of the small intestine. It's going to join with the gastric inhibitory peptide and also be inhibitory to stomach activity. Okay. What's it going to do at the pancreas? going to cause inhibition or release? Release. Which one? Bicarbs or enzymes? Bicarbs. Okay. And what other organ is going to have receptors for secretin? The liver. Okay. So not the gallbladder, but the liver. So it has receptors. Remember, secretin is going to be in the blood as a hormone. So it's going to go to the liver. And the excitatory, it increases bile synthesis. Hepatocytes will make more bile that travels through the canaliculus into the bile ducts and down the common bile duct. Okay. And we're having bicarb ions coming through here. All right, so we have one more, and that's our cholecystokinin. Remember what cholecysto stands for? Chole means, or cysto means bladder. So this can help you keep straight which one goes to the gallbladder, which one goes to the liver. This means movement, all right, so movement of the gallbladder. So it's also going to join the gastric inhibitory by slowing down the stomach. It's going to go to the pancreas. And it's going to be increasing digestive enzymes. So our trypsinogen, pancreatic lipase, pancreatic amylase, those will be increased because of secretin. And then instead of going to the liver, it's going to go to the gallbladder. Now, the gallbladder doesn't make bile, right? It just stores it. So the liver's making bile all the time. It comes down. If this is not open, the sphincter of Oddi is not open because we're not digesting lipid food. It goes back up and gets stored in the gallbladder. And so cholecystokinin causes contraction of the gallbladder. So not synthesis, but release, increased release of bile. Okay. So this diagram is what used to be my extra credit activity. When the digestive system exam had fill-ins still. So next week it's just multiple choice, there aren't write-in answers, but expect, and it's reduced, we only have 3 days of digestive system instead of 4 or 5. So reduced content, but some of that will be on there, okay? So at this point, the mixed chyme, bicarbs and bile and enzymes, are going to move into the next region of the small intestine. And once it's past this knob and past the superior mesenteric artery, what do we call this region of the small intestine? The jejunum, okay? And it's just slightly shorter than the ileum. So we do a lot of digestion and some absorption here. More of the absorption is going to occur in the ileum. And now we're increasing the content of amino acids, simple sugars or monosaccharides, and our lipids. This content is going to go up in the blood. Okay? So we call that the absorptive state. When all those are are being absorbed and their levels of the food stuff is increased. 2 to 4 hours after we've eaten, these levels have dropped, and we call that the post-absorptive state. Okay? So what would be the main hormone that I don't have up on the board but comes from the pancreas for the absorptive state? As we're absorbing our monosaccharides, what's happening to the level of glucose and fructose in the blood? They're going to go up. And as increased levels of glucose, exactly, reaches the pancreas, it's going to increase insulin. Then in the post-absorptive state, as your body has used glucose, and as your body has, under the influence of insulin, taken up glucose, glucose levels are going to fall. So then, when glucose levels fall, what is our major hormone from the pancreas? Glucagon, okay? We have a lot of G words. Gastrin and glucagon and glucose and we're going to see glycogenesis and glycolysis, and might help to make a list of those to be able to keep them straight. All right? So there's a lot of information on there. Again, if you take it hormone by hormone or organ by organ in the sequence in which the food stuff moves through the that first region of the intestine, I think you can help to keep it straight. Okay? And these diagrams on the PowerPoints are not quite as crowded as I have on the board. There's a couple of different PowerPoints for that. Okay. Any questions on that? So now let's look at what happened to the glucose and amino acids and how they cross from the lumen of the digestive tract into the capillaries or lacteals of our small intestine. All right? So the small view of a villus up here has been expanded to show one of the simple columnar cells covering the surface of the villus. And within the fold of mucosa, we have the blood capillary and we have the lymphatic capillary. Okay? So may I erase the board? Can I put that down? Okay. So if we kind of keep things straight, we'll put the muscularis mucosa here. Get an idea of where we are. And then we'll draw a couple of these are supposed to be microvilli. They look like cilia. These would be simple columnar cells covering the surface of a villus. All right. This would be the lumen of the small intestine. So this is where we have our food stuff working. And I'm going to switch. Well, we'll just put in the capillary. And then I'll draw on the lacteal when we come back and do lipid digestion. Okay? So looking at glucose, looking at our carbohydrates, what was the this is a review. What was the intermediate enzyme that breaks down carbs? I heard it, but I didn't understand it. Amylase. Okay? So the intermediate enzyme, by intermediate, I mean the product of this chemical reaction is too large to cross the plasma membrane, okay? So in the small intestine, which has a pH of 8 to 9, because of our pancreatic bicarbonate ions, salivary amylase is no longer working, all right? So what is the intermediate enzyme for bicarbs that works in the lumen? Not bicarbs, for polysaccharides that works in the lumen from the pancreas. Pancreatic amylase. All right, so pancreatic amylase is going to take disaccharides, such as sucrose, it's going to take polysaccharides, carbohydrates, and it's going to work and split them into disaccharides. Okay? So we have sucrose, and we have lactose, and we have maltose as our products. All right, the enzyme that does that activity is found here, within the plasma membrane of our microvilli. So this is our final stage enzyme. So that would be this is what we went over on Tuesday. That would be sucrase, or maltase, or lactase. All right, so our disaccharide has to come very close to the plasma membrane to interact with that enzyme. And when it does, the carbon-carbon bond is broken. Remember the diagram I first drew for you with hydrolysis? So this is going to react with water. And the enzyme, sucrase, the maltase or the lactase, is going to be interacting with that. And the product is going to be a carbon with hydrogen, or a carbon with a hydroxyl group, and we have our two separate saccharides, our glucose or our fructose or our galactose. Okay? Well, that's how we get to this point. Everybody with me on that?
What are the two black circles? I'm sorry.
The two black circles?
Yeah, above them. Yeah, that are interacting with the lactase, maltase.
Black. I don't see a black.
The little circles.
Oh, these are sugars. This would be a glucose or a fructose. These would be the single monosaccharides.
Monosaccharides.
Yeah. So I'll put that there. I was looking for solid circles.
Sorry, yeah.
So that would be the glucose, the fructose, or the galactose that we covered in the previous section. All right. So now, right at the microvilli, we have those monosaccharides, and we need to move them across the simple columnar epithelia and into our capillary of the villus. And that brings us to this diagram, which is a bit overwhelming. Okay? However, this is where we are right here. So this would be a microvilli, just like I drew over here. All right, but I tilted it so I could put the capillary on the side. And before we take a closer look at this, let's add 3 different forms of molecular transport to this cell. So at the bottom of the cell, we're going to have a molecule that uses ATP, and it's going to pump 3 sodium ions out of the cell, and it's going to move 2 potassium ions into the cell. Potassium ions will end up leaking back out again because they're already in there high concentration. So this is going from a low concentration to a high concentration, which is why we need to use active transport. All right, so that's one means of molecular movement. Look familiar? All right. Up here, we have another molecule that is going to allow sodium ion to leak down, to move down by diffusion from high concentration to low concentration, and it's going to bring with it a glucose molecule. Now, this occurs more rapidly because we've lowered the number of sodium ions inside the cell because of our active transport. So what do we call this process then? Secondary active transport. All right, so the sodium will then end up being pumped out of the cell by active transport. How will the glucose leak? Facilitated diffusion. So those are our three mechanisms of molecular movement. So the glucose then is going to move in by facilitated diffusion. All right, and that's essentially what this diagram shows. Lactose and glucose are going to move in via secondary active transport using a symport with sodium, as is glucose. Now, it's saying here that fructose doesn't use that. Fructose comes in by facilitated diffusion, but I'm not going to be picky. All right? So for our purposes, I just want you to recognize that our monosaccharides get in with secondary active transport, set up by the active transport pump of sodium potassium, and they leave the columnar cells by facilitated diffusion. Yes, Caitlin?
When you say set up by active transport, you're saying because the
That
sodium
creates
gradient-
the increased gradient,
Okay.
that creates the drain, that pulls more sodium in than would normally happen. All right? So secondary active transport and facilitated diffusion are actually passive, but the secondary active transport occurs because of the energy used at the pump. Okay? And that's what this complicated diagram is saying. Okay?
Is that going to be like the type of diagram you would show on the test?
There's so many words to take out of that. I'll draw something,
Okay.
but yeah,
So
it'll be
more.
similar to the process. Not the actual one. All right, so same thing happens with amino acids. So we learned on the kidney that we can replace glucose here with a single amino acid. And it would move into the cell by secondary active transport, and the amino acids would move out by facilitated diffusion, all right? So we can add amino acids to this component, but we're going to go through a series of diagrams that help us review the enzymes a little bit more clearly than on that specific diagram. All right, so question number 3. And I'm working on grading the last page of your exam from last week. I have the Scantron to run and total it up, so I'm just grading the last page. So that'll take me about another hour, and I'll get your grades up. I just put the Monday, Wednesday grades up this evening before class. So on that exam, going back to 10 days, what part of the kidney did we see this happening for clearance of glucose and amino acids in the filtrate? Because amino acids and glucose are not normally found in our final stage urine. diabetic with high levels of glucose, all your glucose and all your amino acids will be gone from the filtrate by the time we leave the region. That is the correct answer for this question. Okay, anybody want more time on question 3?
Me too.
Alright, so instead of drawing this up on the board, this is going to be occurring essentially in the same place as what I drew on the board, but let's look at this diagram here. So in the mouth, if we're looking at protein, we have no pepsin equivalent, no trypsin equivalent made by the salivary glands. Okay? So the first time we see that is in the stomach. And pepsinogen is activated to pepsin by the hydrochloric acid. And it's going to break carbon-carbon, it's going to increase the reaction rate for breaking carbon-carbon bonds. We go from polypeptides to the oligo, which means many but not as many as poly. And notice on our protein, each green circle represents an amino acid instead of a simple sugar. And we have a nitrogen end and we have a carboxyl end, our amino acid end. Alright? That's going to be important as we get to the release of single amino acids. So pepsin is going to give us these 5 to 6 or 7 strands of amino acid, which is still too large to cross the plasma membrane, so we have no absorption of protein in the stomach. So pepsin would be an intermediate or a final stage enzyme. Intermediate. We move into the small intestine, and we've added trypsin and chymotrypsin from the pancreas, and they're going to do the same thing. So in the lumen of our small intestine, we can change our intermediate enzyme here to trypsin and chymotrypsin, because the pepsin is going to be degraded. It's not going to work at the alkaline pH. So our intermediate enzyme is going to be trypsin, chymotrypsin. Note that the small intestine doesn't make any of these intermediate enzymes for either carbohydrates or for proteins. It doesn't make amylase, it doesn't make any of the intermediate enzymes for protein. And so our polypeptides, we're going to have some of our oligopeptides already broken and formed in the stomach, and then we're going to do polypeptides in the small intestine. And the small intestine is going to provide us final stage enzymes. So here we have carboxypeptidase, and it's on the amino acid end of our oligopeptide, and it's going to cleave off a single amino acid. Right? The next molecule behind, the next amino acid, that's still going to be the carboxy end. The carboxypeptidase in the lumen of the small intestine is going to just keep on snipping off single amino acid molecules from the carbon end. And we have aminopeptidase that's going to keep clipping off single amino acids from the nitrogenous end. Alright, so those are 2 of our final stage enzymes. We have carboxypeptidase from the carboxyl end. And we have aminopeptidase. And those are the ones that we find in the plasma membrane of the microvilli. So along with the sucrase and maltase and so on, we're going to have these final stage. And eventually, as we're removing an enzyme, amino acid from each end, we get to the point where we have just 2 left. We have the carboxyl end here and the nitrogenous end here, so which way do we go? We use a different enzyme called a dipeptidase that just splits the 2 in half. Okay?
So that's also a final stage.
have single amino acids that will be transported across the plasma membrane of the simple columnar cell via secondary active transport and end up leaving the cell through facilitated diffusion, just like we saw with the glucose or lactose. Okay? And those are going to move into the blood. So the capillary that I've drawn there is found in the villus of the small intestine. And this is part of our first bed of capillaries in our portal system. Okay? So these capillaries join together and carry food away from the small intestine into what vein? Not quite yet. What's the vein that carries blood away from the small intestine? Superior mesenteric vein. Okay? And then that will join with the splenic vein to form the hepatic portal vein. And so our hepatic portal vein and our branches of the hepatic portal vein in the liver are going to be high in simple sugars, or monosaccharides, and high in amino acids, right? But not high in our lipids. So let's look at lipid digestion next. We don't have an intermediate stage and final stage enzyme for our lipids, we just have lipase. So what are the organs that make lipase? Salivary lipase, stomach makes gastric lipase, and the pancreas makes pancreatic lipase. That's the one we're going to be using in our wet lab state. Okay, it's pancreatic lipase. So I'm going to redraw this. If I may erase it now, I'm going to redraw it for lipids. Because instead of having the capillary, I'm going to draw the lacteal lymphatic capillary. Okay. I'm going to take this diagram that's shown vertically and place it into our horizontal diagram. So instead of our blood capillary, we're going to have our lymphatic capillary, which if you recall, simple squamous cells have large gaps, big enough for a whole white blood cell to get through. Make that just a little bit bigger because I need to put in some additional organelles. All right, so we're still in the small intestine. pH 8 to 9. And our enzyme now is lipase. All right? We're working on triglycerides. The form of lipid that we're working on is a triglyceride. And I'll just remind you what that looks like. Triglycerides have a 3-carbon with the associative hydrogen. I left those off. Glycerol molecule attached to a fatty acid, which has a carboxyl group. The fatty acid portion attached to the glycerol, and long chains of carbons which form the fatty acids. So this is the glycerol group, and this is the fatty acid group. All right? Actually, let me move that lower out of the way. I'll rewrite it down here. Yeah, glycerol. And we have our fatty acid. And another glycerol. Another fatty acid. And a third fatty acid. Oh, this is a triglyceride. All right, these are fatty acids. And this is what we're going to be working with pancreatic lipase during lab today. So we're going to be adding a solution called pancreatin, which has a whole bunch of different enzymes. But we're focused on the pancreatic lipase. And when we, well, hold on 1 second, and I'll show you. So this is the whipping cream that I purchased yesterday, or actually Tuesday. And it has a powdered litmus solution added to it, as I mentioned before. The purple color, or lavender color, is showing indicating the pH is alkaline. This is the pH around 8.5, all right? So that's the pH at which our lipase works. And when we release fatty acid, what's going to happen to that pH? The fatty acids are going to release hydrogen ions, and the increase in hydrogen ions will change this solution. It'll become more this color, well, redder purple color, all right, showing that fatty acids are being released. But we have an issue with fats. Oil and water tend not to mix very well, and so if we have fat in our diet, it tends to form large fat globules, and even though we twist it and turn this in the stomach, we tend to see large fat globules will come back together if we don't use bile salts. All right? So these salts are in the bile that was added from the gallbladder and from the liver. And so what happens is the small lipid droplets, the bile salts, like detergent, have a hydrophilic and a hydrophobic component. So the hydrophobic inserts itself into the lipid droplet, and the hydrophilic interacts with the water and the lumen. And the bile salts repel each other, so when they happen to come up against another one, instead of joining with them, they move away. All right, like two positive ends of a magnet. So when we have these smaller lipid droplets with the bile salts, we call that structure a micelle, M-I-C-E-L-L-E. Okay. Now the lipase is able to work on this because there is there's surface area available, and so as the lipase interacts with the lipid, with our triglyceride, it's going to release, it's going to work on the strain of electrons between the carbon and the oxygen group here. And we're going to see monoglycerol, which is 3 carbons with 1 fatty acid, and the other 2 fatty acids separately, because they're lipids, they're going to be able to move directly through the phospholipids, between the phospholipids. And they're going to come into this cell as a 3-carbon molecule. As our monoglyceride, okay? And as our long-chain fatty acids. Because they are lipid soluble. But they don't stay that way. After all this trouble to get them into smaller molecules that can cross into the plasma, cross into the cell. They move into the smooth ER. Remember, smooth ER never binds ribosomes. It's functional in detoxification, plasma membrane synthesis, and steroid synthesis. So these move into the smooth ER, where we reform the triglyceride. Okay? Now, it's still not soluble in water, but we don't have our biosalts anymore. The biosalts got left out here in the lumen. So we have to figure out, biosalts are soluble in the water because they have a charged end. Remember, water is polar, covalent. So we need to cover the lipid with something that is also polar covalent. So the triglyceride from the smooth ER pinches off and attaches to the Golgi, which has proteins from the ribosomal ER. Not bad to end up talking about Bio-430 again. All right, so now this is in the Golgi, picking up proteins. Picking up proteins from the Golgi. Proteins are polar covalent. They have charges. All right, so proteins, as we know from the liver, are soluble in the blood. So this pinches off from the Golgi. And it contains our lipid with its protein coat. Any idea what those proteins are? LDL, VLDL, HDL, heard of those before? But now, since it's coated with the protein, it's too large to, it's no longer lipid soluble, so it can't lead through diffusion. So it fuses, this membrane sac from the Golgi, fuses with the plasma membrane of our cell, and the protein-coated lipid, which is too big to get into the capillary, now moves into the lacteal. Okay? So we call this structure, lipid with a protein coat, is known as a chylomicron. Okay, so my cell out here where it's the lipid with the bile salts, chylomicron here after it's coated with the protein. We go through this whole process of breaking it down and reforming it and then coating it with another protein to get it out. So, the chylomicrons that are in the lacteal, are they going to go into the portal vein? Are they going to get into the superior mesenteric vein and the portal vein? Where does our lacteal go? You remember from the lymphatic system. It leaves through lymphatic vessels and ends up in the cisterna chyli. And the cisterna chyli empties into the thoracic duct, which empties into our left subclavian, and then our brachiocephalic, and superior vena cava, the heart. After it gets back from the lungs, the aorta, the abdominal aorta, celiac trunk, common hepatic artery, and goes into the liver beside the portal vein, but in the hepatic artery. So our hepatic portal vein is going to be high in sugars and amino acids, and our hepatic artery is going to be high in lipids and oxygen. And our liver uses both. That's why they flow into the liver side by side, which is unusual, you remember. Normally you have an artery going in and a vein going out. okay? So the portal vein and the hepatic artery go in together, and we have the hepatic vein sweep. Alrighty? So it's a few steps here, but not as complicated. You actually break it down and think about what's happening. We just do one thing and then we do it backwards. Alright, so as our food moves into the large intestine, remember the cells there are 25% mucus. So our purpose in the large intestine is primarily absorption, but not of digested material. That's finished up in the ileum. And in the large intestine, we're absorbing vitamin K. It's being made by the E. coli that are breaking down the fat fibers that we can't break down, and some other fat-soluble vitamins. Okay? And that and water is essentially what we get back from the large intestine. So as we get to the rectum, we're back to the same sequences of sensory stimuli and activities as we saw first with reproductive, and then repeated with bladder. So is defecation reflex going to be a parasympathetic process or a sympathetic process? Parasympathetic. It's part of homeostasis. So stretching of our reflex fibers is going to be carried by sensory neurons through what nerve? What carried stretching of the bladder? Sensory neurons, stretching of the uterus, for that matter. Sensory in the pudendal nerve. All right. When that gets to the sacrospinal cord, it's going to form a reflex with parasympathetic, which is going to come back through the pelvic splanchnic nerves, and going to increase peristalsis of the rectum. Just like the increased contraction of the urinary bladder for voiding, it's going to increase peristalsis of the rectum. What's it going to do to the internal sphincter, that smooth muscle? Is it going to relax or constrict? It's going to relax, okay? Which, unless you're not potty trained, I mean, if you are potty trained, you have to then inhibit the external anal sphincter. If you're not potty trained, then you're going to defecate and hopefully have a diaper on. So what nerve innervates the external anal sphincter, which is rectal? Sorry, which is skeletal muscle. Pudendal. All right. So when we activate the pudendal nerve, is the external anal sphincter going to contract or relax? It's going to contract. If you want to defecate, you have to inhibit that pudendal nerve going to the external anal sphincter. Okay? Because parasympathetic and sympathetic will they release acetylcholine or norepinephrine, depending on what cell they bind to, will either activate or inhibit. So we can release neurotransmitters and cause relaxation. That's what the acetylcholine does with the heart, okay? As far as the essay notes. That's what acetylcholine does to the the internal urethral sphincter. Parasympathetic acetylcholine binds to receptors on the internal urethral sphincter and causes it to relax. Same thing with the internal anal sphincter. Acetylcholine binds to receptors on these smooth muscles and causes them to contract. So they can actively do both. However, to get the external anal sphincter to contract, we don't release acetylcholine from the pudendal nerve. We have to stop releasing it. We have to inhibit that. And that starts in our brain. Okay, that's a conscious decision. All right? So, are we at 5 or 6? 4. Did I release some here? Great, that should be 4. Okay. Oh. So, this is, did I show this to you guys or Monday, Wednesday, or both? Did I show this to you guys?
We know what it is.
Okay, it's yes, the ad for the potty squatty. And this is unicorn ice creams coming out of his butt. Puborectalis is part of the levator ani. I figured this was nicer than putting actual, I don't know, they make collectible poop now. Plastic. So, which of these is occurring? Which of these is a true statement? It's a little while to read, so I'll give you some time. get a little tired of seeing the ice cream come out in.
C looks like it. C looks like it.
Move them. So is muscularis externus smooth muscle or skeletal muscle? Smooth. That's our inner circular and outer longitudinal, which, except for our internal anal sphincter, is limited to the tinea coli. Okay? But the internal anal sphincter is all smooth muscle. There's some word I changed it to save it. So innervation muscular this result is by no, there's less than one. Yeah, sorry. Mark all that are correct here. There's one more. There is more than one correct answer.
Oh, great.
How many correct answers are there? I had this question last night. One of the students said, "So how many correct answers are there?" And I can't say because it's like giving out everything away, right? So remember I said pelvic splanchnic is going to do two things. It's going to increase peristalsis and it's going to relax the sphincter. Okay? So increasing peristalsis is A. That's what's going to move the fecal matter down the rectum. Sympathetic inhibits that. Well, norepinephrine is binding to receptors, alpha beta receptors on the rectum. It's going to inhibit peristalsis. That's true. That's B. Right? Lower motor neurons are in the pudendal nerve, and when they release acetylcholine, that causes the external anal sphincter, which is skeletal muscle, to contract. That's true. All right? To set up this whole reflex in which you do defecate requires increasing the amount of fecal matter in the rectum, especially the lower rectum, which stretches the rectal wall and excites stretch receptors, which sends sensory neuron impulses back to the spinal cord to excite the parasympathetic nervous system via the pudendal nerve as well. Okay? So sympathetic is in sacrosplanchnic, parasympathetic is in pelvic splanchnic, and sensory and voluntary motor is in the pudendal. Okay, so all four of those end up being true statements. All right, so let's take a short break, and when we come back, we'll look at a little bit of actually, let's do the quiz. So go ahead and take your break, and this will be the histology quiz. So take some study time for that, and let me know when you're ready. The histology of salivary glands, histology of our GPAP before we did it on the models, mucosa, what's the submucosa, what's the muscularis externus components. Be able to identify an organ by its histology, so the pancreas or the liver, the parts of the liver, bile duct, central vein, branches of the hepatic arteries, and then just the different parts. You know, is it a pancreas? Is it a liver? Is it a small intestine? Is it a colon or whatever? And cells of the stomach, so the parietal cells, the endocrine cells, the chief cells. So there will be a couple of model questions on the liver and pancreas components. Okay. Maybe large intestine, but only those.